to be able to kind of identify what it is that you're targeting and what it is that you're wanting to demonstrate change with respect to interventions that you're introducing becomes very, very complex, which brings us onto the topic of biomarkers, which is incredibly complicated. And I think also, we need to consider the fact that biomarkers, they are critically needed to be able to, for us to be able to conduct that clinical research, but it's certainly not simple. And we know that biomarkers need to be validated, and to validate biomarkers, it requires clinical evidence to demonstrate that a change in that endpoint, i.e., a surrogate biomarker, then is also correlates and predicts a specific clinical benefit. So, when we're thinking about reduction in epigenetic age, which is a very exciting field and something that you are very well versed in, it's only meaningful if it's associated with a positive clinical change. So we really need to understand about whether this change in epigenetic age and any changes that are happening at the molecular and even the cellular level are able to be translated at the macroscopic level, and ultimately a benefit that's translatable to the patient or the consumer. And so this is something, very much thinking about when we think about, bringing in a topical or injectable product for skin aging. We really need to drill down to what is the benefit to the patient, rather than something that we can talk to that's at microscopic level, for example. And this will be related to both efficacy measures, but I think also very importantly, patient-reported outcomes and potentially observer-related outcomes, especially in the aesthetics field. So this is where you're always needing to make sure that you're correlating back to what's meaningful for that person who's getting that treatment, who's getting that intervention. And then another big challenge, which we cannot forget, is the regulators. And so you need to gain regulatory approval to get these interventions approved on markers. So therefore, there's a high bar to be able to provide the evidence to the regulator that that biomarker is reliable and repeatable and consistent, and especially it's suitable for the intended purpose. And so all of that needs to be taken into account, and we can't neglect the fact that we know that the aging process is not unilateral and is not consistent across demographics and populations. So we need to take into account that phenotypes vary according to skin types, ethnicity, sex and gender, geographical location, and that includes exposure to pollution and UV light. So all of that, and it's and I know that this is a big undertaking that we're all trying to tackle, but we need to understand all of these different factors before we can actually then demonstrate that we're having a clear benefit in relationship to the actual aging process of individuals.